So we're going to move on now to another group of organisms, so another domain. All right, so we are looking at the domain Archaea. Archaea are another group of prokaryotes. At one point in time, they were just grouped with the bacteria as prokaryota, and then also as bacteria as a group. And now bacteria are their own domain, and Archaea are their own separate domain. This came about proposed in 1977 by Carl Woese. And so the late 70s and into the 80s where people started to accept it a little bit more. And to be honest, at this point, people still think of it as being new, that the Archaea are their own group, but they have been now for many, many decades. So one of the problems with them is that the difficulty in studying the Archaea. So the Archaea, while they are prokaryotes in that they do not have a nucleus, and they're small, so we think of them as being like the bacteria. But they actually have a number of qualities and specific characteristics that make them actually more like the eukaryota than the bacteria. And so that's what I'm going to go over now. Is not everything. I have a different lecture for in a microbiology course. You could look it up if you were interested, to actually go through the names of the groups and all within the Archaea, but that's not what we're doing in this class. Part of the problem is, and it's going to be the case with this particular group, with the Archaea, it's also going to be with the Protista, which we'll get into after this, is that I'm going to focus more on characteristics, because those characteristics are the way they are. We know this is, the particular structure of the phospholipid that's unique, I'll talk about here in a second, for the Archaea. Or this is a particular type of flagella structure, or these sorts of things. They're actual characteristics of the organisms. Now, the names of the organisms, they are constantly being changed all the time. They've changed from 10 years ago to just 2 years ago to maybe even this year. Some different groups are being proposed and people are trying to rename things. So to have you memorize names of groups that are constantly changing and will probably continue to change, there's not a lot of point to do that. But we're going to focus on characteristics, things that are not really going to change. These organisms will have these characteristics, and these are the unique features of the characteristics. I will talk about the names to some degree because you need to know some of that for communication, to ask questions, and when you're reading, you need to recognize some of those names, but generally that's not going to be the focus. So if you're interested in that, go ahead and look at it. If you're not that keen on it, then it's okay. I'll only point out what you need to know. So, Archaea, what makes them different or unique? All right, so we're going to start off with, and we'll kind of keep some of the same flow of organization, which I'll be using for a variety of the different groups to kind of standardize the information a little bit, and how I'm going to talk about it. So one of the things I'm going to talk about here are the phospholipids. So, Archaea, these are single-celled organisms. Single-celled organisms, all organisms that are cells or are made up of cells, the cells have a membrane. Okay, so the cell membrane here is unique. How is it unique? Well, a typical cell membrane is made up of phospholipids. All right, and so this is a general structure. So the phosphate functional group, a glycerol, so that's phosphate, glycerol, and then fatty acids, so 2 fatty acids. Again, we're not in a chemistry class, but the characteristics of the organisms rely on you understanding something about the chemistry. That's what's making them different or unique. So we have to talk about it a little bit. You don't have to necessarily draw these structures, but you should know and be able to point out the names of the characteristic features and explain something about these particular features, which is what I'm going to do now. So this is bacteria and eukarya. So us, plants, fungi, the protists, and all the bacteria have this kind of phospholipid structure, right? And so there's a bond here, and this is one of the things. The bond that connects the fatty acids to the glycerol. This type of bond here is referred to as an ester bond. Now, if we look over here for the Archaea Phospholipids, they have the phosphate functional group, they have a glycerol, and then they have the fatty acids. But there's going to be some now changes to this. Okay, so here we have different arrangement, right? The oxygen here is bonded to carbon and this carbon. You don't see this one, so this is called an ether bond. And you don't have to know the detailed chemistry of it in a class like ours, but just know that one difference. What's one difference in the cell membrane that makes it unique? The Archaea have an ether bond in the phospholipid structure as opposed to the ester bond. That's just one part of it. Second part, these phospholipids that have the fatty acids are just chains of carbon. So it's carbon to carbon to carbon to carbon to carbon in a chain. They can be full of hydrogens, like this. And so every little bond on the side here will all be hydrogens. And they could be straight. That's called saturated. Or they can have double bonds between the carbons and be unsaturated, and that causes bends and changes the shape. So there can be variation here. These are not exactly the same. There's different lengths, put like some dots here, different lengths to the carbon chains, and they can be saturated, unsaturated, polyunsaturated. What makes the Archaea different is that the Archaea have side chains, so they have other carbon branches attached to them. There's all different kinds of varieties, patterns of these. So there's not just one set for this as well. So the Archaea have branching side chains to the phospholipids. So ether bond, branching, I'll just refer to as the fatty acid chains. That's another characteristic. So those deal with the chemistry of the phospholipids themselves. So we'll look at the membrane. Now, generally, a phospholipid, to really simplify it in terms of its structure, has a polar head. So this whole area up top here is polar. It likes and or interacts with water. And then, draw this, jump ahead a little bit here. And then we have tails, right? Fatty acid tails, like this. So that's these two things, sticking off the polar heads. Now, in bacteria, protists, fungi, plants, animals, all the eukarya, okay, we have a phospholipid bilayer, like this. So the fatty acid tails associate with fatty acid tails of another layer of phospholipids, but it's inverted. So you kind of have water, on one side interacting with the polar heads, water on the other side interacting with the polar heads, and then you have a non-polar barrier. Okay, so that's where the tails are. In Archaea, so now here's the difference. What we see most of the time is that these tails are linked. So we don't have a bilayer. Now it's called a monolayer. But you have to keep in mind, that's what you'll read. It has a phospholipid monolayer. Well, a monolayer then implies, one layer of phospholipids, but here you have the phospholipid with essentially two heads, one on each side because they're fused phospholipids. So in a way, it's the double layer isn't just separate individual phospholipid molecules freely moving around, which is going to affect factors like fluidity, which you might study again in a cell biology course or other course. But here the tails are fused together. There's going to be some reasons for that. We talk about roles, ecological roles, like where we find the Archaea. And one of the reasons why it's difficult to study the Archaea is because many of the Archaea, not all, but many of them are extremophiles. That means they live in extreme conditions. So essentially the conditions that we would say, oh, nothing could live there, that's too hot, that would kill any living thing. But we find archaea live in these environments that are incredibly hot. We could say, or that environment is too cold, or that environment is too salty, or that environment has certain poisonous chemicals in it that are poisonous to pretty much anything that's alive, and nothing can survive there, except then we end up finding, oh, look, there are things that live there, and they're archaea. They're these organisms that live in those environments. So they have to have special, unique adaptations, biochemical adaptations in their cells to live in these environments. The cell membrane isn't the only adaptation for all these things, but it's definitely one of the things that can help them at high temperatures. So at high temperatures, what would happen is the phospholipids would pull apart from one another, the membrane would become weak and fail, and the cells would die. But because they have this fusion of the tails, it just makes them stronger. And so their little cell membranes have a little tougher time than being torn apart at high heat. And they're going to have other adaptations as well, protein adaptations and other sorts of things. But these are one of the sort of biochemical cellular modifications that they have. And we're kind of, so you would ask, but why? But why do they have that? This isn't necessarily the answer, but it's part of the answer is to adapt to some of these extreme conditions and allow them to live there. So quick, coming back to archaea, what's some of the differences that they have? Well, the first real differences has to do with the membrane. There's not just a single difference. So we have a couple, we have ether bonds, we have branching side chains, and then we have fused phospholipids, which we call monolayer. My pen is dying, but you can kind of see here monolayer, and that's what we have here. I already wrote it down here, monolayer. So that's the cell membrane. And then we can get into all kinds of chemistry differences in it. And again, it's not really for an organismal class like this one, but we're just trying to talk about some of these things that you may just see a throwaway sentence without a real explanation of the detailed what is it or why it might be that way. And so I'm just trying to give you a little bit of that additional information. Next, single-celled. They typically have a cell wall. Now, again, here's where we're going to find big differences. Okay, so bacteria have a peptidoglycan cell wall. That peptidoglycan structure is NAM and NAG sugars, these two specific sugars that repeat over and over and over again. And then there's peptide or amino acid side chains that are fused together. The difference is really between, there can be all kinds of different peptidoglycan, and that the side chain composition can vary to make up sort of different compositions there. But it's pretty much the same structure over and over again in all different sorts of organisms, gram-positive and gram-negative. And differences between those are how thick they are. So the gram-positives having a very thick peptidoglycan layer, the gram-negatives having very, very thin layer. What you will read is that while archaea have cell walls, there's all different kinds. So the cell walls, first off, are diverse. There is no one type of cell wall in the archaea. You will read and find things that say they do not have peptidoglycan. However, they have structure. If you were just to look at it, drawn with just like circles and squares, it would look just exactly like a peptidoglycan layer. And in fact, it's sometimes called pseudopeptidoglycan or pseudomurin because it's very similar. One of the main differences is really just different sugars in the chains, and then they can be held together with the peptide side groups. So we call this pseudomurin, which is essentially a variation of peptidoglycan. So similar to peptidoglycan. Again, if you were to actually look at the structure in a very generic sense, it would look very much the same. If you look at it on a detailed chemical analysis of what are these sugars and the specific way in which the bonds are formed, like what carbons are bonding for 1,4, 1,6, those types of bonds, you're going to find the differences. But generically, pretty similar structure. Now, but that's not going to be for all of them. So that's some, so it's diverse. Some of them might have this pseudomurin structure. Some of them have no structure like peptidoglycan whatsoever. Nothing at all. They don't have the pseudomurin. They have nothing else. But they have to have something to kind of help protect the cells not only from the outside but from their own internal pressure. So I talked a little bit about this with the bacteria. The internal pressure without internal membranes, which is going to be the same thing here, no internal membranes, no nucleus. That's why they're prokaryotes. It's not just the lack of a nucleus, but the lack of the internal membranes. Without that, they can have a high amount of osmotic pressure. So one of the ways that they can compensate for that is with something outside the cell membrane to hold it together, to keep it from bursting. The membrane itself, we can see now, is going to be different, a little bit tougher in some cases, but they still need some kind of cell wall. Bacteria, many of them will have something called an S layer. It's sort of a protein shell. Some of the bacteria have that, but actually many of the archaea have it, so this is actually fairly common. And it could be in addition to the pseudomurin layer, or it could be in place of it. So there's nothing like the peptidoglycan pseudomurin layer, there's just this S layer. So essentially outside the membrane is a protein shell, and that's it, and it's this S layer. They can also have different types of sugar structures, complex branching sugar structures, which without going into like the detailed, just call it branching carbohydrates layer. Because there's a whole variety of different ones based on the carbohydrates and the bonding and other things that they're attached to, but it can be an outer protective layer of carbohydrates that are protecting these cells, forming a cell wall type structure. And again, these can be cells that only have one of these, or they could have multiples of these. And then the chemistry of them can be very unique and very different if you were to go into really the detailed levels of the chemistry. So they have cell walls, but they have great diversity to them in terms of how many they have, the types of materials that make them up, and how they differ then from other organisms, especially the bacteria. All right, so those are some of the, we got the cell membrane making the cell wall outside the cell. What else? So internally, they're going to have a lot of similarities between the bacteria and even us. So they'll have DNA, the DNA basic structure is the same. The proteins, ribosomes, pretty much kind of going to be the same as ours. Now, with some of the processes we'll talk about as we look at things like DNA, structure, replication, and gene expression. Now, for all living things, there are very generic ways in which these processes occur that are going to be similar. So DNA uses the template to make pieces of RNA. That's the same for all living things. All right. The way in which the RNA or the ribosomes work to assemble the proteins, very similar. Transfer RNAs with amino acids come in. There's codons in the RNA that will match up, and then they'll assemble the amino acids with peptide bonds. So these are all cell biology details, and generically, once again, the same. But if we start to look again at the details of like, what are the actual enzymes? Who does this? What is the order of some of these events? We start to find that there are more similarities between the specific enzymes involved in these processes and the uniqueness of them to eukaryotes than there are to bacteria. So the enzymes in these processes more similar to eukaryote proteins, so the enzymes that are involved in DNA replication and the gene expression process. In addition to that, so also as a subset, a characteristic of the DNA of our cells and eukaryotes in general have structures called introns. So introns are regions of the DNA that don't directly code for pieces of RNA. So a long time ago, people thought they called them junk DNA, but now they realize they have functions. Some of them are regulatory in function, so they're binding sites where proteins and things bind, even though they don't code. In addition to that, some of them are evolutionary holdovers, so from ancestors. So it's ancestral DNA still in the DNA. It's nothing that's actually expressed, but it was there in the past, and then it's carried on through the new organisms that come about. We don't see that in the bacteria. So bacteria have no introns. Their genome, their DNA, just codes for, everything codes for something. But in the archaea, they have introns. So their introns are then like our introns in some ways. Okay, so that's another similarity that puts the archaea being a little closer to eukaryotes than they are to bacteria. As far as motility goes, in movement, not all will have flagella, but they can have flagella. In the previous lecture, I talked a little bit about the prokaryote flagella. In the protists, in the next lecture, we're going to go into actually a lot of detail of the eukaryotic flagella. So this is a topic that kind of you should have some idea of to compare. So essentially compare the flagellar structure of the bacteria with the archaea with the eukaryote flagella. So, and what's different about each of them and unique about them. So here, the flagella is made up of unique proteins, not the flagellin protein that we find in bacteria, and not the tubulin proteins that we find in eukaryotes, so it's its own unique proteins. The flagellum moves in a slightly different way. It does spin, but it spins in an opposite way of the bacterial flagella. So it has sort of an opposite spin. So you could take away from that it does have a spin. Our eukaryotic flagella don't spin. They move back and forth like this, like the way the cilia move, and then the flagella, because they're longer, tend to have a more undulating movement. But the bacterial flagella spin like this because of motor proteins. The flagella of archaea spin as well, but in a different way. They have similar, but again, different motor proteins, and the way in which they use energy to create that movement is different as well. So, different energy source. All right, so again, without going into all these details, but to mention some of the differences, I have to point some of these things out. So they can move, but not all of them are going to move, and we'll see that. Reproduction, typically a binary fission, but they can do other things as well. So again, bacteria do binary fission. Which is cell division, where essentially one cell divides into more two cells, and those two cells are exactly the same as the others. But archaea can do some other things as well. They can do something called budding, where essentially a smaller cell kind of like buds off of a larger cell. Well, not larger, it's just the normal cell size, but essentially just you get two cells, but they're not exactly the same. This new cell here, well, they're both a little bit smaller than the original, but this one is much smaller, for example, and then it has to essentially grow, whereas that doesn't really happen as much in the bacteria. They kind of divide, and there does have to be additional cell wall added and cell membrane added, and you got to increase things, but it's not as different as this. You find the budding process, which we do see this in protists. We see this in yeast and fungi, the type of budding process as well. So they have some different ways also in which they divide their cells. So one of the main things, really the main thing that divides up this group into subgroups, so often referred to as the different phyla within the archaea, are genetic sequences. Just like with the bacteria, specific sequencing of ribosomal subunit genes that are then used to say these are more similar, so they go into one group, and these are different. They go into another group, but they're similar to each other, and that's kind of the main way. As far as those groups, what do they do that is unique, right? And so there are some groups, for example, and I'm not going to put on everything from all of them, just a few noteworthy things. They are the only known, not all the archaea, but there's a group of archaea that are methane producers, and they're the only known living cells that produce methane. So bacteria do not produce methane, and eukaryotic cells do not produce methane. But there are archaea, not all, but there's groups of archaea that can produce methane, which is important for carbon cycling and a variety of other environmental processes as well. So that's something that's very unique to this particular group. There are others that can that are photosynthetic, and they can do it in slightly different ways in which than the prokaryotes or plants do photosynthesis. In fact, some of the pigments used in photosynthesis for these organisms are more similar to animal proteins that are found in our eye. So the pigments that absorb light in our retina are actually more similar to the particular pigments in archaea that are used in photosynthesis. So what we see here is that there are biological characteristics, biochemical characteristics that we could find in certain groups. We could find them being very different in other groups that seem related, and we could find them being very similar in groups that seem completely unrelated. So it makes it kind of confusing in trying to use those characteristics as any kind of link, except to say maybe there's a common ancestor with some of those characteristics, and some organisms kept those and continued to have them, and other organisms lost them or didn't continue to express them, which is kind of where the introns also come in. Genetic information carried from ancestors and then might be used in the same way or in different sorts of ways. So again, as extremophiles, we'll kind of come back to this. This is one of the reasons why it's difficult to study. So even over a fairly long period of time, relatively little is known about the archaea specifically because they're difficult to, say, grow in a lab and culture, because the conditions that they live under, people cannot live in those conditions or work in those conditions. So you have to create an environment inside a laboratory to potentially grow and work with these organisms, and that is a great challenge in many cases, especially to keep it and maintain those conditions, so without creating something very artificial for them. So studying extreme survival at extreme pH, extreme temperatures, pressures, so deep in the ocean, salt concentrations. There are halophiles that live in extreme salt concentrations, and that's just the thing, is to keep something in mind. It's not only that they can tolerate these extremes of pH or extremely high temperatures or salt concentrations that would destroy and kill other things. It's that they have adapted to it where it's a requirement. They require those conditions. If you were to bring them to normal just water without salt, fresh water, they would die. If you brought them down to our room temperature, which usually refer to organisms that live in the temperatures that we live at, it's called mesophilic, meso meaning kind of like middle or mid-range, and they would die. Now there are some that are not extremophiles. Like I said, there are some mesophiles that like the same temperatures that we do. There's some archaea that live in our own bodies. Right? So they're not only found in hydrothermal vents in the bottom of the ocean or very bizarre salt lakes and places like that. They could be found within us and within other animals, and associated with plants and associated with our soils, and they're all really all over. It's just that many of them are these extreme groups. So finishing up, archaea, a whole separate domain of life that relatively little is known about compared to the other groups. What do we know about them? They're single-celled organisms. They're considered prokaryotic cells because they don't have a nuclear envelope or other membranes inside the cell. As we look at their membranes themselves, they're made up of phospholipids, like ours, yet different in the chemistry. So unique types of bonds, ether bonds, unique differences in the fatty acids where they're branching structures. There are unique ways in which they're joined together to form the bilayer. Instead of being two separate layers, they can be fused into a single layer. Their cell walls can be very diverse and unique compared to plant cell walls or nothing like them, except in role, their role for the organism. And biochemically, some similarities between the bacterial peptidoglycan, but it's chemically different, and again, much more diverse. Genetically, they share the basics with all living things, but their mechanisms of some of the processes with gene expression and replication are more like the eukaryotic processes than the processes in the bacteria. And then as their movement is unique, with the flagella having unique structures and the way in which the structures work. Physiologically, that's where they become greatly diverse and incredibly unique. To go into all the unique things they do to adapt to these environments is a whole sort of separate thing that can be focused on, which could be very, very interesting. But again, it's not something we're doing with our particular class, just kind of introducing the group. And so as I said, to finish up, if you are interested in looking at some of the names of the groups or how the groups are organized or what are common characteristics, there's a separate microbiology lecture I have just on the Archaea, classifying them. But that's not something you have to worry about in our particular organismal class.